So I'm actually going to go into the next section some, just to keep it moving. That's from us.
Is tomorrow going to be the 3.2 still though?
Yeah, tomorrow's still 3.2, but we might be halfway through it today. Does that make sense? Are we filming?
Yes, sir.
Yay!
Oh no, look at that nice cheese.
People at home, if you're wondering why I have an eye patch on under my shield, it's because I was in a bar fight with 3 guys, and they were all bigger than me.
But you were in the hospital.
They won. No, I don't know. I scratched my cornea. Isn't that crazy? Do y'all know what a cornea is?
It's eye part.
It's the clear part of the eye on the outside. And I don't know how I did it. I just woke up with it. It's the craziest thing. Yes. Hurts really bad. Feels like there's something in my eye. Have you ever felt that?
I got hydrogen monoxide in my eye a few days ago.
I've had a double stack.
Alright, so enough about me. Let's talk about food. You ever eat food?
Oh yeah, sometimes. All the time actually.
Food contains the 4 types of organic molecules. Carbohydrates, proteins, lipids, and nucleic acids. And that's what you have to learn, these 4 types of organic molecules. You have to understand their structure. You have to understand what they're for. You have to be able to recognize them and point them out and say, oh, that's a lipid. Oh, that's an amino acid. Oh, that's maltose. Could you do that now?
No, thank you.
No, you couldn't. That's why we're going to learn. Now, the first thing we need to understand is how these things are built. Now, a monomer is a single unit. It's just, the word monomer is a general term for a single unit. The word polymer is a term for a bunch of single units put together to make a larger unit. So this is a monomer. That is a polymer. You see the difference? Monomer is 1 unit. Did you know mono means 1?
Yeah.
Have you ever played Monopoly?
I have.
1 guy owns all the property at the end. Have you ever worn a monocle?
Yeah.
I wish I could.
That's a lens, a single lens. It's kind of neat that the Monopoly guy has a monocle. You ever notice that?
Yes, it's quite funny.
There you go. A polymer, poly means many. Polymer. So poly means many. You have many monomers hooked together to form a polymer. Make sense? Now, how do they hook together? We need to know that. Excuse me. Having a rough few weeks. Come on. Plus, our remote control mouse isn't working. Whoa. If you take, can I just not see this? You'll see, is there a thing on the screen? You see it?
It's that, yeah.
Yeah, there it is.
Oh, there it is.
No, that's at the top left.
As far as the...
Alright, I got it. Sorry, it's hard to work on my control mouse. Let me show you a dehydration synthesis reaction. Can you say dehydration synthesis reaction?
Dehydration synthesis reaction.
Wow! I'm impressed. That's way better than the other biology classes.
Do we get extra points?
I'm going to tell them how lame they actually are.
Me too.
All right, so here is a glucose molecule. Here's another glucose molecule. If they come together, they're going to hook together. And I don't know how well you can see this. Maybe I'll turn the light on. Maybe you can see it a little better with the light. But some of y'all are sitting far away. But this glucose has an OH right here. This glucose has an OH right here. What will happen is an enzyme, usually, will come up and will grab each of these. An enzyme is like a protein. Y'all remember enzymes from biology?
Yes, sir.
It'll grab each of these and it'll put them together. And this H will come off and this OH will come off. And now we have HHO. You know what that is?
Water.
H2O. That's water.
That's the piece I did.
So we form, by joining these together, we join those together, these come off, we form a water molecule. That's why it's called dehydration synthesis. Dehydration, have you ever been dehydrated before?
Yep.
Oh yeah.
Dehydration is the removal of water. So it's a dehydration, we remove water, it's a synthesis reaction. Synthesis means you're joining two things together. So we're joining two things together by removing water. Dehydration synthesis. Does that make sense? What's that?
Where's the other oxygen?
The other oxygen is now in between the bonds. So there's a bond going to this oxygen, and this has a bond going to that oxygen. So if you look, there's...
My bad.
Hold on. So if you look at this, do you see... You know, I can't use the freaking pointer because I only have one arm. You see that oxygen right there?
Yeah.
So that's that oxygen. That's that oxygen right there. Who asked that?
Oh, I was just gonna say, isn't it because oxygen can make two covalent bonds? Or is that a
Yes, oxygen can make two bonds, and there they are, 1, 2.
I'm
Great
so
job.
God, I'm smart.
That's right.
Wow, look at you
You're
guys.
genius, Carter.
Now, another thing you need to understand is when they draw molecules like this, this is a shorthand version of the actual molecule. Do y'all know that carbon can have 4 bonds?
I did.
Heck yeah.
You should know this. It was on the last quiz. Carbon can have 4 bonds because it can share with 4 other things. It has 4 outer level electrons. So, right there, at that spot, they don't write the C. They assume that you know that.
At the bend, like the little dents.
Thomas, are you looking? You seem like you're looking at the ground. Right there is a carbon atom with 4 bonds. 1, 2, 3, 4. But they don't draw the carbon. You're just supposed to assume that. You assume there's a carbon there. 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 Do you assume there's a carbon there?
No.
You don't, because that's an oxygen there. They drew the oxygen for you.
That's so kind.
Can you look at this molecule and come up with the formula for glucose?
Sure.
How many carbons are there?
6.
There's 6. So that's C6. How many hydrogens are there? Let me tell you something about the hydrogens. Do you notice that there's a bond going up here that's leading to nothing?
Yes.
They assume that you know that that's hydrogen. Why do they assume that? Because they assume things. I don't know.
It'd be messy.
But it's too messy if you write in all the hydrogens. So they leave some out, and you just assume that if there's a bond going to nowhere, that it's going to be a hydrogen. So why didn't they just leave that hydrogen blank? Why couldn't they just have a bond going to nowhere? I don't know. I didn't draw the thing. But they could do that. And I know they could do that because they left one there, you see?
I do.
And you just assume it's to a hydrogen. Are y'all understanding the shorthand way of drawing these molecules?
Yes, sir.
Now sometimes they'll just do this. I've seen glucose represented like this.
Just
And you assume that it's glucose. How do we know that's glucose?
Because the oxygen.
Just because it is. I don't know. That's
You know.
just know that. Glucose is a pretty commonly drawn organic molecule because it's
Sugar.
ubiquitous. Do you know what ubiquitous is?
Absolutely not.
Like universal?
Common in a lot of things. Seen often. Yes.
We're gonna do that little like stick and ball thing lab.
Yes. We're going to build some of these in the lab. That's why I want to go ahead into the next section so we can spend some time in the lab building these things. Does that make you sensey?
Sensey making.
Are y'all good to go on dehydration synthesis? Oh, there's another term for it. It can also be called a condensation reaction. Condensation. Have you ever heard that word?
Yeah.
If you look on the window, if you breathe on the window, you'll see your breath. That's condensation. It's air, it's water molecules sticking to something. So since this reaction produces water molecules, we call it a condensation reaction. We good? And by the way, if you hook one glucose molecule to another glucose molecule, you now have a new molecule called maltose. That's maltose
What's maltose?
and water. That is a maltose molecule.
So glucose plus another glucose plus water?
No.
What?
Glucose plus a glucose makes maltose, and water is released.
Oh, okay.
Water comes out of it. Make you sensey?
Sensey making.
Now, this is the opposite happening. This is taking a larger molecule, adding water, and it breaks it apart. It's called a hydrolysis reaction. So, watch this. Here's my big molecule. Let me go back, lights on for this. Here's my maltose molecule. Let's say I wanted to break it apart. I could take water and add water to it, and it would break apart. The H goes on one molecule, the OH goes on the other. Did y'all see that?
Glucose?
What's that? Yeah, it goes back to two glucoses. So here's water, here's maltose, that's what we start with. Here we go, it connects and whoop, breaks apart. And now the water is part of the two glucose molecules. We call that a hydrolysis reaction. That's called hydrolysis. Hydro means water, lysis means to break. To break using water, hydrolysis or hydrolysis. So, have you ever had like crusty stuff all over your dishes and you set them in the sink in a bunch of water? And it breaks up the molecules, doesn't it?
It does.
You give it some time. What's happening is your food are big molecules like this, and water is coming in, and it's attaching to the ends of these molecules and breaking them apart. And that's why you do that. It's washing dishes. I just explained washing dishes. Now you know. Soap makes it break up even faster. Soap breaks up fat molecules. And so, soap really helps too. Soap and water, and you're washing dishes. This is what you can do with your life. You can wash dishes the rest of your life.
Sweet.
Make
What
minimum
joy that
wage.
would be.
There you go. Maybe you can shoot for that. The grades in here don't work out too well. Okay. You can tell the people in the kitchen, you can say, hey, I'm doing a little hydrolysis over here. Little hydrolysis reaction.
You can sound so smart.
Yeah, and they'd be like, ooh, this dishwasher's sharp. Much sharper than the last dishwasher. You want to see video footage?
I did.
Heck yeah.
It's not anyone watching.
And I was like, man.
Is this what I think it is?
Many important biological molecules are made from hidden subunits called monomers.
Whoa.
When many monomers join, the
Hello?
result is a polymer.
Stop
For
talking.
example, amino acid monomers join to form a protein polymer. And glucose monomers combine to form a complex carbohydrate polymer. Biological polymers form by dehydration synthesis reactions. As you can see here, each of the monomers in this reaction has a hydrogen, or H, and a hydroxyl, or OH group. In the course of the reaction, the hydrogen is removed from one monomer and the hydroxyl group from the other. The hydrogen and hydroxyl group combine to form water, and a bond links the two monomers. Hydrolysis is the opposite of a dehydration synthesis reaction. During a hydrolysis reaction, a polymer is reduced to its monomer subunits by the addition of water. In fact, the word hydrolysis literally means to break water. The hydroxyl group from a water molecule attaches to one monomer, and the remaining hydrogen attaches to the other monomer. In other words, water is used to break the bond holding monomers together. Let's do a quick recap. During dehydration,
Let's not.
Alright.
There was a very famous experiment done in the 1950s, I think, where scientists knew that these molecules can attach to one another and build up and form something more kind of more complicated. So a guy named Alexander Oparin, he was a chemist, a Russian chemist, and he made a hypothesis. He said maybe life developed this way. And why this is important is because this is biology, the study of life. Maybe living things formed from simple molecules attaching to one another and forming something more complicated. So that was called Oparin's hypothesis. Well, time went by, and a couple of scientists, gosh, there's names slipping my mind now. Somebody Google life in a test tube experiment. It's two scientists. Starts with an S.
Miller and Urey.
No, it doesn't start with S. Yeah, Miller and Urey. Miller and Urey were two scientists who wanted to test this experiment. So they set up this apparatus that looks like this, and they took some of the molecules that were present in early Earth's atmosphere, like water and methane and ammonia and hydrogen. Those were present in early Earth's atmosphere. We know this from studying old rocks. There are rocks on Earth that are billions of years old, and you can break those rocks open and study the gases that come out of them. Those were the gases that were there when when those rocks were forming billions of years ago. So we know that these molecules were present in Earth's early atmosphere. What if we put them in a chamber, we put them in liquid form, we heat them up, they boil, they turn into steam, they go in this chamber, and let's put some electric impulses in there, shock them a little bit, give them some energy, and see what happens. And lo and behold, these things, after just running into one another, see, molecules, when they run into one another, they often stick by this dehydration synthesis type reactions. If they hit each other hard enough, they'll stick and form something more complicated. In just a few hours of doing an experiment like this, you start to build up complicated organic molecules like this on the walls of the container. And indeed, what they do is they let it run through here and cool it off, and run through here, and they suck some of it out, and they sample it, and they found out that there were just after a few hours of this experiment, there are chains of sugars, polysaccharides. There were amino acid chains, small proteins being formed. There were nucleotides, the building blocks of DNA had formed. And there were lipids, fats. The same lipids that surround each cell membrane had formed after just a few hours. So that is certainly not evidence against that hypothesis. The operant said, hey, maybe life could form if you let these things combine, and sure enough, some of the same molecules you find in living cells are forming right there in just a short amount of time.
Why don't you just let it go on for a while?
They have. As a matter of fact, they've been running this experiment for like 70 years now, and they have formed things called protocells that look and behave almost exactly like cells. They have a lipid bilayer, they have enzymes in the middle of them, they absorb substances from their surroundings, they grow, and when they get big enough, they divide in two, just like cells do. The only thing they have not developed is a DNA molecule on the inside of these things. They've developed short strands of nucleotides, but not the long DNA strand that actual cells have. So, but they've only been doing it for 70 years. It probably took life about 300 or 400 million years to actually evolve. So maybe if you kept running that experiment for millions of years, you might come up with something like that. That's the idea. Of course, they want to come up with it faster than millions of years. We don't have millions of years to see where you make something like that. So they have ways of running this experiment faster by heating it up and stuff like that. I predict, I don't know this for sure, but I predict you will see a cell form in a lab before you die. A totally new evolution of a cell that's separate from the life we have now. And that would be one of the biggest news stories in the history of science, a new form of life that is not...
Get ready, because Teenage Science is here!
Wow!
Generation G.
Get that.
Now, how did the molecules of life arise?
That's Carl Sagan, my favorite.
In a laboratory at Cornell University, we mix together the gases and waters of the primitive Earth, supply some energy, and see if we can make the stuff of life. But what was the early atmosphere made of? Ordinary air? If we start with our present atmosphere, the experiment is a dismal failure. Instead of making proteins and nucleic acids, all we make is smog, a backward step. Why doesn't such an experiment work? Because the air of today contains molecular oxygen. But oxygen is made by plants. It's pretty obvious that there were no plants before the origin of life. We mustn't use oxygen in our experiments because there wasn't any oxygen in the early atmosphere. This is perfectly reasonable because the cosmos is made mostly of hydrogen, which gobbles oxygen up. The Earth's low gravity has allowed most of our original hydrogen gas to trickle away to space. There's almost none left. But 4 billion years ago, our atmosphere was full of hydrogen-rich gases: methane, ammonia, water vapor. These are the gases we should use. Taking great care to ensure the purity of these gases, my colleague, Bishankari, pumps them from their holding flasks. was first performed by Stanley Miller and Harold Urey in the 1950s. The starting gases are now introduced into a large reaction vessel. We could shine ultraviolet light on this mixture, simulating the early sun. But in this experiment, the gases will be sparked, as the primitive atmosphere was by early light. After only a few hours, the interior of the reaction vessel becomes streaked with a strange brown pigment, a rich collection of complex organic molecules, including the building blocks of the proteins and the nucleic acids. Under the right conditions, these building blocks assemble themselves into molecules resembling little proteins and little nucleic acids. These nucleic acids can even make identical copies of themselves. In this vessel are the notes of the music of life, although not yet the music itself. Now, no one so far has mixed together the gases and waters of the primitive Earth and at the end of the experiment had something crawl out of the flask. There's still a great deal to be understood about the origin of life, including the origin of the genetic code. But we've only been at such experiments for 30 years. Nature's had a 4 billion year head start. Incidentally, there's nothing in such experiments that's unique to the Earth. The gases we start with, the energy sources we use, are entirely common through the cosmos. So chemical reactions, something like these, must be responsible for the organic matter in interstellar space and the amino acids in the meteorites. Similar chemical reactions must have occurred on a billion other worlds in the Milky Way galaxy. Look how easy it is to make great globs of this stuff. The molecules of life fill the cosmos.
My hero. Okay, so that's the end of the section. Let me just go into the next section. We'll finish it next time. The next section starts on carbohydrates. We'll do carbohydrates first, and then lipids, and then proteins, and then nucleic acids. That's this whole next week, or this whole week is what we're doing. One of these each day. So we start with carbohydrates. And normally I show carbohydrates in this ring form. But they don't have to exist as rings. They can exist as straight chains. This is what glucose would look like if you straightened out the chain and had it in a straight line. But glucose tends to form a ring, and this type of glucose, the straight chain form, you actually don't see as often. It will kind of curl around on itself like a belt. You can straighten out a belt, and then you can curl it around on itself. And there's all different types of carbons. There's 6 carbon chains, and 5 carbon chains, and 3 carbon chains. There's all different types of carbohydrates. But we'll just be focusing on a few of them, glucose being one. Another one we'll focus on is called galactose, and another one is called fructose. All 3 of these molecules have the formula C6H12O6, which means they're isomers. They're the same formulas. Now, I have here in my models here, and we're going to, notice I have them on every board. We're going to be going over to the boards, and I'm going to have you identify these. We're going to do that tomorrow. But this is glucose. Glucose looks like this. And here's also glucose. It's a hexagonal ring. It's got a CH2OH group sticking up. And we call it alpha glucose if the two hydroxide groups here are going down. And we call it beta glucose if one's going down and the one on the other side's going up. So there's alpha glucose and beta glucose. Now, this molecule right here is called galactose. There's glucose. There's galactose. Can you tell the difference between the two? There's one difference. This OH group, or HO group, it's a hydroxide group. It's sticking up in the same direction as the CH2OH group. Might be hard to see from where you're sitting, but if the OH is sticking up in the same direction as that, it's galactose. If the OH is going down, as it is in both of these, away from the CH2OH, it's glucose. And that's just a tiny difference. Galactose, they're both going up here. Glucose, they're going down, away from the CH2OH. That small difference is the difference between some enzymes being able to break down the molecule and other enzymes not being able to break it down. It's just a small difference, but it's important. And then fructose looks like that. That's fructose. And that's easy to tell because it's a pentagon. Even though it's still got 6 carbons, there's a CH2OH group here and a CH2OH group here, and it's a 5 carbon. It's a pentagonal ring. So this is galactose, and this is glucose, and this is alpha glucose. My alphabet. This is alpha glucose, and this is beta glucose. Y'all know Greek letters? Alpha, beta, gamma, delta, epsilon, zeta, theta, kappa, mu, xylophone, rho, sigma, epsilon, phi, psi, omega. That's the Greek alphabet. I had to be able to say it 3 times while holding a burning match to get into my fraternity in college. Why? Still don't understand why. Yep. Lit a match. I'm going to get rid of those ones I did. I'm going to use them. I'm going to get rid of those ones I did. And you say it 3 times. And if you didn't, you'd burn your fingers. And that'll teach you. I don't know if it ever taught me anything. We call each of these molecules monosaccharides. If you hook together 2 monosaccharides, you make a disaccharide. Monosaccharide, monosaccharide, put together, disaccharide. Glucose plus fructose, do you know what that makes?
Sugar.
Sucrose, which is table sugar, the same thing you put on your cereal in the morning. You load it up, don't you? 7-8 teaspoonfuls. Already on top of the frosted cereal you began with. And yeah, that gets you going.
Marco.
What do you think about all of this? I went through some of the next chapter there for you. We're going to pick up there next time. We got new wipers. Do y'all know we got new wipers? Who are the new wipers? Let's see. Meryl and Josie did it last time. Josie and David. Is there anybody absent? Is everybody here? Yeah.
Everyone's here, I think.
I forgot to say, everybody pack up. And Josie and David, you're wiping.